Okay, thank you very much, and thank you to all the organizers. It's really been a wonderful meeting, and I would like to have you students organize all the other meetings because it's actually worked much more efficiently than most of the ones I've been to. So today I'm actually not going to tell you about basal ganglia. I'm going to tell you about something completely different, which is a small project in the lab, and I thought it would be kind of fun to change a little bit what we're talking about. So this is not going to be a very molecular talk. I apologize to those of you that want molecular talks. And it's not going to be a grand overview of what my lab does, but rather sort of a focused presentation of one problem that is one that I find very interesting and kind of fun to think about. And so most of my lab, the work in my lab is motivated by trying to understand this. How does an animal choose what to do next? And if we think about our own behavior, we tend to overemphasize really reasoned behavioral choice. So as humans, we like to think that we do a careful survey of all the information that's available to us, that we think hard about what we want to do, and then we make an informed choice, for example, where to go have dinner. And of course, these choices are based on our past outcomes, where we've eaten before, what we've enjoyed, our sensory environment, right, what we're in the mood for that day, some internal state, and our goals. Now, that's one kind of action. In fact, most of what we do during the day is actually governed by a different kind of process. Much of what we do consists of automated, goal-oriented, and learned motor sequences. And so that's very easily exemplified by somebody playing a video game. Of course, this is a completely unnatural situation. This man is interacting with these artificial input devices in order to navigate through an artificial world in which he collects very esoteric rewards, in this case consisting simply of points. Yet, this kind of behavior really outlines much of what we do during the day, including, for example, speech or typing on our keyboards or getting up from our desk to go get a drink from a soda machine, for example. That kind of behavior depends heavily on the basal ganglia, and most of my lab studies the basal ganglia. We try to understand the properties of synapses and the modulations of synapses within the basal ganglia, trying to understand how their regulation contributes to shaping behavioral choices by animals. The basal ganglia are phylogenetically very old, and they're evolutionarily highly conserved. Their importance to human behavior is made very clear by diseases of the basal ganglia, which have very strong behavioral effects, including, of course, Parkinson's, Huntington's, and so forth. So this is most of what my lab does. And today I'm going to tell you about another kind of behavioral choice. And so when we were thinking in general about this question, how does an animal choose what to do next, we wanted to find a behavior that we could study that was innate, that we didn't have to teach. It was quite easy to monitor. It's conserved in its importance across evolution, that can really be described as binary, and that actually reflects a decision and not a reflex. And so this was a problem that was taken on by two graduate students in my lab, Helen Ho, who's now graduating, and Min Suk Hyun, who's continuing on with this project. And so I thought it'd be fun to talk about this because this is entirely a graduate student-driven project in my lab. And so the behavior we chose to study is actually what's shown here. Okay, the release of urine. And so those of you who have dogs know that this is not really a trivial process. This animal spent a long time investigating its environment. Despite possibly a great urgency to release urine, it may have been in your house for 10 hours, 12 hours without going outside. It still navigates the environment, carefully considers many different spots and trees and so forth, and then decides to release urine in a particular place. Now, the release of urine is technically called micturition. And it's important to realize that this micturition is not simply a reflex. Okay? It really reflects an that's spelled wrong, ethologically important decision that's influenced by many factors, including the experience of the animal, its sex, its sensory state, social hierarchy, its goals, and so forth. And of course, I'm outlining here a dog, and I'm going to show you data from mice, yet all of these things are still relevant for humans. I mean, all one needs to do is look at the increase in public urination that happens with alcohol consumption. So clearly, once you de-repress these central sensors, then it's much more favorable to go and pee outside. So I'll tell you just a few facts about micturition to make you understand why I think this is an interesting problem, and then tell you a little bit about what we're doing here. So the first thing to realize is quite important, which is the spinal cord reflex does not actually drive the release of urine. In adults, spinal cord reflexes actually cause urine retention. So the more your bladder fills, the more your spinal cord works to retain urine. And this is why in adults who have, for example, an acute spinal cord transection, for example, following a motorcycle injury, and a medical emergency that develops is the inability to release urine. So the release of urine in an adult is really driven by the brain, and it's driven by de-repressing that spinal cord reflex. So every time you release urine, this is something that's coming from your central nervous system that's overriding a peripheral reflex. Now, mice also highly control how they release urine. These are some classic experiments that were done by Desjardins, as you see here in the early 70s, in which he placed two dominant male mice side by side in this arena that had a solid partition between it. So these mice could not interact. They didn't know that the other ones were there. He allowed them to roam around for a while, then took out some paper that he had placed on the bottom of the cage and simply asked where was urine released. And you can see that each of these dominant males released urine in these large spots, mostly located around the periphery of the cage. Now, something very interesting happened when he replaced this fixed barrier with a permeable barrier. You could see that now the mice who could detect the presence of each other one suddenly changed their pee pattern dramatically. Now they released urine and scattered it in these small dots throughout the entire arena. So this simple, very simple manipulation of simply letting one mouse know that the other mouse was there dramatically changed its pee pattern. Okay? And of course, this is the pattern of a dominant male mouse that's trying to mark and capture territory. Now, there's also something very interesting that happens if we look at a dominant male and a subordinate male. Again, in exactly that same arena, interacting for the same amount of time, they can't actually touch each other, right? So there's no aggression here. They simply know that the other mouse is there. One is dominant, the other one is subordinate, and you can see the male dominant, again, vigorously marks the territory, possibly now even increasing the number of marks that it puts here near the divide, whereas the subordinate goes and pees in the corner. Okay? And so this is the typical pee pattern of a subordinate male mouse. It's also the typical pee pattern of female mice in most conditions. So here we have a very, again, simple manipulation, interaction. Clearly, pee patterns are driven by the sensory environment of the animal, by its social hierarchy, by its past experience. So we wanted to understand how does all this information about the environment and experience impinge on this circuit. And we wanted to be able to trace from the periphery all the way to the motor output the complete sensory motor transformation and therefore have a circuit dissection of this simple decision. In order to do this, which is an ongoing process, we're certainly not done with this by any means, we decided to break this problem down into 4 steps. One, we had to establish a behavioral assay that we could run reliably in a laboratory setting on laboratory mice. We then needed to find the neurons in the circuit that control the bladder, find the input to these neurons, and eventually trace the flow of activity through this circuit, and again, understand its transformation from where it represents sensory environment to where it represents a motor decision. So let me take you through those steps, and this is what Helen and Minsook have been working on. So here is the assay that we've typically used the most. We have 2 male mice that are housed together within a cage, and so they establish a social hierarchy. One becomes dominant, the other one becomes subordinate. And if now we remove them from that arena and place them in another arena in which we have a filter paper on the bottom and put a little bit of saline just in the center of it, you can see the pattern that we've seen before, a little bit less robust. Here, the subordinate male pees in the corners, and the dominant male here pees a little bit more generously throughout the arena. Now, if we take the same males and instead expose them to a little bit of urine of an estrus female mouse, you can see now this quite dramatic pattern. So the subordinate male, again, simply pees in the corners, doesn't really care about the estrus female urine, whereas now the male mouse adopts this vigorous pee pattern that occurs throughout the whole cage. And so this is the assay that we're going to rely on heavily. Simply an odorant here can now change rapidly the pee pattern, but of course interfacing somehow with social hierarchy, these 2 things must be combined. Now, there's a couple of things that we needed to prove to first understand whether this was an interesting problem or not. So one possibility is that the animal micturates in one spot because that's where it spends more time. If this were true, this would really not be a very interesting problem to study because it would say that possibly animals simply release urine at a constant rate during all time, and if the animal spends its time here, well, obviously that's where the urine is going to be. And so in that case, we would be studying a problem of navigation as opposed to one of controlled micturition. Okay, again here, possibly the subordinate spent all its time in the corner where the dominant male ran all around. So is this true? So in order to study this, we decided to build a pee tracker system, and this consists of an arena. It's a transparent arena in which we have a mouse, and there's a filter paper at the base. And we have two ways to monitor the mouse. The first is through an infrared illumination system on the top. We use that to track the location of the mouse. And then from the bottom, we have a blue illumination system, which causes the urine on the paper to fluoresce. And so here we can, in real time, watch the marks of urine, and here we can see where the animal is. So here's such a video in a square arena now. You can see the female urine in the center, and help you appreciate that oftentimes when the male mouse interacts with that urine, it leaves a little bit of a mark of urine here. And I think one can tell, at least anecdotally here, that the mouse spends a lot of time in the edges, but yet tends to pee only when it's interacting with this central spot. Okay? And so this argues that place occupancy and micturition are distinct, and therefore one can independently study one decision without the other. And here's a summary from a bunch of mice. You can see that on the left, this is a heat map that shows us where the animal spends time, and most of you that work on mice know that mice tend to stay near the corners and the edges of arena, whereas here is the distribution of pee. Clearly these are very distinct, and this is a high frequency event that occurs despite the animal not spending that much time in the arena. Okay, so what are the circuits, therefore, that could control the bladder from the brain? There was some classic work that was done by Frederick Barrington, now quite some time ago, that identified a region in the pons way back in the brain, and he identified this in cats by doing lesion studies, that eventually became known as the pontine micturition center. In the classic literature, you'll see it referred to as Barrington's nucleus. And what he identified was that if he lesioned that part of the brain, that a cat would still try to urinate. It would still adopt the posture of a cat trying to urinate, but would be unable to release urine. And so he reasoned that this was a part of the brain that must directly control the bladder. So what is this pontine micturition center? Here you can see from an atlas this very small nucleus labeled with a B for Barrington's nucleus. It juxtaposes the locus coeruleus. It's quite small. We've done a lot of analysis of this, and I'll show you some of the numbers in a minute. It's about the same size in a mouse as in a human. It's a little bit bigger in a human, probably arguing that they really have quite similar function in the two. It's not a part of the brain that expands tremendously in a primate, most likely because we don't use urine to mark and for social communication that frequently. Here is the Barrington's nucleus seen in a different view. This is a mouse that expresses Cre recombinase under the promoter for the cortico-releasing hormone. Many of you know that CRH is a hormone that's present in a lot of stress-regulated pathways within the brain. It plays an important role in triggering the release of stress hormones in the periphery. In the brain, it's also found in pathways that are related to stress, and it's included expression here within the pontine micturition center. So this animal has been crossed with another mouse that expresses green fluorescent protein in a Cre-dependent manner, and so therefore it lights up the nucleus, and we can see it quite clearly. Here's another view of the same nucleus. Now you can see in purple the same cells that express this cortico-releasing hormone, and in green we've stained with tyrosine hydroxylase, stained for tyrosine hydroxylase, an enzyme that's involved in the production of noradrenaline and dopamine, and you can see very nicely the Barrington's nucleus here nestled against the locus coeruleus, which is the major noradrenaline producing part of the brain, also located in the pons. So we can find this nucleus in acute brain slices as well as in fixed preparations. And it turns out that this nucleus contains about 1,000 neurons on each side, and about 500 of them express this cortico-releasing hormone. We've performed electrophysiological analysis of the cortico-releasing hormone producing cells. The details of that don't matter too much for here, but they have this characteristic sort of slow firing rate, whereas the non-CRH expressing neurons look more like typical GABAergic interneurons, fast firing rates, and so forth. And we've done an analysis by in situ hybridization of the genes that are expressed by these cells, and it turns out that these CRH expressing cells express markers for glutamate packaging and glutamate release, whereas they don't express enzymes that are involved in the production of GABA, an inhibitory neurotransmitter. So based on this kind of analysis, we reasoned that these neurons may be the glutamatergic excitatory neurons that send an output to the sacral spinal cord and to the bladder. In order to look at that directly, we made a virus. We made an adeno-associated virus that encodes placental alkaline phosphatase, whose expression is dependent on Cre recombinase. And so we injected that virus into the mouse that expresses Cre recombinase in these CRH positive neurons, and then simply asked where can we find the enzymatic activity of the alkaline phosphatase. So here you can see the injection site. on this side of the brain. You can't really see it on this monitor, but there are some processes that are revealed that go to the other side of the brain. So there's bilateral interactions between the pontine micturition centers, as well as this extensive ramification locally. But most importantly, if we then look at a section of the spinal cord in the distal cord, you can see bilateral innervation of the sacral cord in the location where we know are the ganglia that then innervate the bladder. So indeed, these neurons from the brain are very long-range projection neurons that send their axons all the way down to the sacral cord to control bladder function, so we believe. So these neurons, as I mentioned, are excitatory projection neurons, and so we reason that they might be the command signal that drives bladder contractions in response to different signals coming in the brain. If so, then their activity should correlate with and be sufficient to trigger bladder contractions. And so at this point, we turn to a series of approaches that are not common to my lab, but are ones that are sort of inherited from classical physiological approaches to study organ function in animals. And we learned how to place pressure transducers inside the bladder of these animals to directly monitor the contractions that were going on there. We coupled this with an approach that Carl Dieseroff talked about on the first day, which is fiber photometry. And so we expressed a calcium-sensitive green fluorescent protein called GCAMP in these CRH neurons, again using the same trick of exploiting Cre recombinase and Cre-dependent adeno-associated viruses, and then inserted an optical fiber into the brain of the animal near the pontine micturition center and used a laser to monitor the fluorescence that was being emitted by these cells. And so here you can see the bladder contraction transients that were measured by this pressure transducer in the bladder, and you can see this very typical pattern, which is that as we infuse saline into the bladder, the pressure begins to build. There are these little contractions of the bladder which don't actually release urine, and then there's a large contraction of the bladder which drops the pressure below the baseline that it had before, indicating that there's been successful voiding, the pressure drops, and then the bladder starts to fill again, contractions and so forth. Okay, so this is what's going on in the bladder. You can see the slow time scale. And now we can look at the fluorescence transients from here reporting calcium influx into these cells, and you can see this very nice correlation where these cells become active, have calcium influx that coincides with these large contractions. And if we simply align then what's going on in the brain to the start of the bladder pressure transients, you can see very nicely that these cells show activity that starts a little bit before the bladder contracts and then outlasts it for quite some time. And this is an interesting behavior that we're still trying to study. We don't really know whether the dynamics that we see here are filtered too much by the slow calcium indicator, or whether this actually reflects the time course of the contraction of the bladder, something that we're trying to study. So that was in an animal that had a pressure transducer in its bladder under anesthesia. We wanted to understand if an animal that's normally exploring its environment and releasing urine is also have this tight correlation between the activity of the CRH positive neurons in its bladder. So we used, again, this P-tracker setup that I showed you before. Now there's no bladder pressure transducer. We allowed the animal to roam around and again measured signals from the brainstem from these neurons. And so you can see here, these small transients in the activity of the CRH neurons and these large transients. And if we look at the video of the animal, we can see that at the time of these transients, if we look at the video frames before and after, that there's deposition of a new large urine spot. Okay? So in an animal running around, once again, this correlation holds. And if we simply, once again, line up the times at which the animal releases urine, we can again see these transients indicating activation of these cells within the brain. Okay, so now is the activity of those cells actually sufficient to trigger a contraction of the bladder and a release of urine? Here again, we use the tricks that Carl Dieseroff talked about at the beginning. We again used a Cre-dependent adeno-associated virus, and this time to put channel rhodopsin, the light-activated cationic channel, into these CRH positive neurons, and once again implanted an optical fiber into the brain of the animals near the pontine micturition center. And so here you can see that with each one of these blue light pulses, we can induce a transient in the bladder, and such that activation of these neurons is actually sufficient to induce bladder pressure and contraction. And here you can see another view. This is the animal that expresses channel rhodopsin. When we turn the laser on, we can then align these pressure transients within the bladder, and you can see that in some cases, but not all cases, we're able to elicit these activations and contractions of the bladder that are seen here on average. Okay, and this does not occur in an animal that lacks the channel rhodopsin. So these neurons then, as I mentioned, are glutamatergic. They're output neurons. Their activity seems to be the P command signal. And so now the question then is reduced to one that's what governs their activity. Where do they get their information in the brain about sex, hierarchy, environment, bladder state, odors, and so forth? And remember, these are only about 500 neurons. Okay, so it's a very small collection of neurons that needs to be integrating information about many different contexts and very different snapshots of what the history of this animal has been. So the goal now is therefore to track from this pontine micturition center back to the sensory organs, for example, to the nose, where it will receive information about the presence of other males or of estrus females. And so here we used an approach that was developed by a variety of people, but really pioneered by Ed Calloway at the Salk Institute, which was to exploit the ability of rabies viruses to move retrogradely across synapses. And so many of you may know that infection with rabies is really devastating to the nervous system. And the reason for that is that once one neuron becomes infected with rabies, it can make a lot of rabies, and that rabies then hops to all the presynaptic partners of that neuron and therefore spreads through the nervous system very quickly. Because each neuron can make so much rabies, it eventually ends up killing the cell, and then you get a severe neurodegeneration. Now, this can be harnessed to exploit, to measure the circuitry within an animal by putting in two tricks. First is that the rabies is rendered incompetent to move across synapses by deleting its G protein. So the G protein is necessary to make a functional rabies that's able to exit one cell and enter another cell. So if the rabies is deleting its G protein, it can't move across synapses. The second trick that's used is to change the way rabies enters the cell to make it require expression of a particular receptor on the cell surface in order to enter the cell. And that cell surface receptor is called TVA. Okay? So now what we do is we make two viruses, two adeno-associated viruses that encode different proteins that are expressed in a Cre-dependent manner. One is TVA fused to M-cherry. Of course, this is then the receptor for rabies entry, so we can direct the rabies to enter only the CRH-expressing neurons of the PMC. And the second AAV encodes that G protein that I mentioned. And so by putting the G protein within the CRH-positive neurons, we allow rabies, once it's entered those cells, to move to the presynaptic partners of that cell. But the rabies itself does not encode G protein, therefore once it moves to the other cell, it's unable to then move again to another cell. Okay, so we both allow rabies to infect these cells and then to move one hop. And so it's a relatively complicated experiment. We first inject the AAVs into this part of the brain, then a week or so later, we come back and inject the rabies. We come through a different path into that same nucleus to prevent accidental co-expression of these things in a different part of the brain. Then we wait several weeks, and then in a nice collaboration with Paul Oudson at Cold Spring Harbor, we then serially cut the entire brain and process it all, looking for parts of the brain in which we can now find GFP representing retrograde expression of the rabies. And so here you can see the infection site. We started from one of the pontine micturition centers on one side of the brain. You can see the nice green expression as well as the M-cherry indicating the site of infection. And now we can analyze through the entire brain to understand where are the other regions that are projecting here. And so we can find, remarkably, a very large number of neurons distributed throughout the brain projecting, as I mentioned, to these small collection of only 500 neurons. And so, for example, we can find inputs coming from far in frontal cortex, where we think information about social hierarchy may be represented. We find inputs coming from areas within the hypothalamus, for example, a pre-optic area, as well as from other very interesting parts of the brain, such as the bed nucleus of the stria terminalis, and so forth. And so we've cataloged all of the inputs that are coming into these cells throughout the entire brain. And as I mentioned, it's on the order of about 30,000 neurons that are labeled by this approach, which is probably quite a severe underestimate of the number of cells that provide input to this collection because of the low efficiency of the rabies transsynaptic hop. So clearly this very small nucleus, despite having only 500 cells, is getting a lot of information from different parts of the brain. Now I'm going to show you just a little snapshot of what's happening in the pre-optic area. This is a part of the brain that we are very much interested in because previous studies had, oh sorry, I'll skip it, I lost it there. So the pre-optic area, previous studies had shown that this part of the brain was necessary for male aggression, this modulated male aggression against other males, whereas studies in females had suggested that it played a role in maternal care of the offspring. So it already seemed to be involved in some kinds of social interactions, the kinds of interactions that might modulate micturition behavior. So what are the kinds of cells in the pontine micturition center that send projections from the pre-optic area that send projections to the pontine micturition center? Min-Suk in the lab developed a very nice in situ approach where he could detect rabies-infected cells by in situ hybridization and then look at other transcripts. And so here, for example, you can see that these rabies-infected cells express the enzyme responsible for GABA production from glutamate. And so, in fact, these cells that send the projections to the PMC are GABAergic inhibitory neurons that are inhibiting that nucleus. And as I mentioned, this is a brain area that shows differential gene expression in social context and contributes to male aggression. So is this part of the brain actually responsible for and contributing in some way for this kind of regulated micturition behavior that I showed you in a minute? Here we exploited yet another trick that neurobiologists are using quite frequently, which is not optogenetics, but what we often refer to as chemo or pharmacogenetics. And so here we expressed a protein that's called HM4D. This is a protein that was made and designed by Brian Roth's lab at the University of North Carolina, which consists of a muscarinic receptor, a G protein coupled receptor, that's been mutated such that it no longer binds acetylcholine, which is an endogenous ligand in the brain, but instead is activated by a small molecule called CNO, clozapine N-oxide, that can be delivered in the periphery. And so in this way, we can express this G protein coupled receptor in particular cells. It's inactive at base, but then when we deliver this small ligand to the animal, the G protein coupled receptor is activated, and it actually shuts down the activity of a population of neurons. And so in this way, we can reduce the activity of particular cells over a period of time. And so we injected this Cre-dependent adeno-associated virus into the pre-optic area of a mouse that expresses Cre recombinase under the promoter of the gene that encodes for the vesicular GABA transporter. So all GABAergic neurons express this gene because this protein product is necessary for packaging GABA into neurotransmitter-containing vesicles. Okay. And you can see here the behavior that results. So on the left is the normal P pattern that we see. The dominant male vigorously marks the cage. The subordinate male here just pees in the corners. He peed so much here that he actually destroyed the filter paper there, but you can imagine that it was there. Now, what happens when we give clozapine N-oxide to these animals? Well, you can see the behavior of both of these is perturbed. They start now peeing in these large, much larger spots. But more importantly, the differences between the dominant and the subordinate male disappear. We've quantified this in a variety of different ways, but here's simply one metric, which is looking at the urine mark size. You can see that normally the subordinate makes larger marks than dominant, but now those differences disappear. And there are various other metrics by which one can quantify this, and this goes away. So this suggests that the GABAergic neurons of this area, of the pre-optic area, contribute to this rank-dependent marking behavior. Now that's actually as far as we've gotten in this project, and I realize that I must have spoken very quickly because I'm done in only about half an hour. But we're tracing back from the pontine micturition center, as I mentioned, into the nose, through the olfactory bulb and through the accessory olfactory bulbs. I've shown you a little bit of data about what happens when we manipulate the pre-optic area. We're now putting fibers and electrodes into this part of the brain to understand when these cells become active in this kind of behavior. But what I like about this project, it's exciting, is that we quite literally only have about 3 synapses that link from the input areas in the brain that detect sensory information about olfactory state to the output to the pontine micturition center. So only with 3 synapses and 3 intervening neurons, we have, I think, a good potential to follow signals from the olfactory or through internal state all the way to the output and fully understand this transformation that happens from sensory to motor command signals. And so we're going to look for where does not only the olfactory information come in, where does that information about social hierarchy, about the sex of the animal and the state of the bladder come into play using the kinds of tools that I've talked about so far. Okay, so that's all I have to tell you about today. I mentioned some of the students along the way. Helen is the one who pioneered this project in the lab, and it's been taken over more recently by Min-Suk Hyun as Helen graduates. Kiwi Wang is also a very talented student in the lab who makes the rabies viruses, which he learned to do in Rob Malenka's lab at Stanford when he was an undergraduate. This kind of project requires a lot of double and triple transgenic animals in which they're virally manipulated and so forth. We really have a fantastic support staff in the lab that helps make these things possible. And the serial tomography and reconstruction of the brains for rabies was done by Pavel Ustin's group and his postdoc Julian Taranda at Cold Spring Harbor. And we had some very important help from the Department of Medicine at the Beth Israel Hospital, in which there are experts in bladder function and so forth that helped guide this project. So, thank you.